Hola. Hola. No, no. Ah, ahora sí. Ostras, sí que va con retraso. Vale. En teoría creo que sí, pero no sé. Creo que sí. Bueno, cuando queráis, yo qué sé. Claro. Hola. Ya, ya. Me siento como marciano. Creo que sí, pero ahora no lo veo. Creo que sí. Lo de la gente online no lo sé, pero antes me ha dicho Rebeca que había gente esperando. Lo hago en inglés, ¿no? Bueno, no, no, da igual, da igual. Sí, bueno, ya. Sí, ya lo hago en inglés ahí. Well, thank you, Mayalen, for introducing me. My name is Àlex Giménez. As Mayalen said, I come from the Institute for Cross-Disciplinary Physics and Complex Systems in Palma de Mallorca, where I'm doing my PhD thesis under the supervision of Manuel Matia. So I was invited here to come here at IFCA for a month. I'm staying here for a month. Indeed, today is my last day. and I was invited by Jose and Mayalen. First of all, I want to thank them and the other members of the group for their hospitality. I've really enjoyed my stay here, and I hope to come back at some moment in the future. So today I wanted to show you a little bit the things that we are doing. This collaboration started because we developed this climate-driven epidemiological model for species of grapevines, and then we thought of integrating this model with climate change scenarios. So let's start. First of all, just to explain you a little bit what is this disease. This is a vector-borne plant disease that affects grapevines. It's caused by a bacterium that is called Xylella fastidiosa, and it has this name because it is very difficult to work with in the lab, and then it's very fastidious, and this is the real reason. And well, as a vector-borne disease, it is transmitted by some vector, in this case, spittlebugs that are a kind of insects, like this one that you can see here in the bottom. And well, we can see here in this map that this disease is native to the Americas and it is already endemic in the US. But recently, there have been some introductions of this bacterium in Europe. Not all of them have led to establishment of the disease, but in some places, like Mallorca, for instance. So this has raised a lot of concern in the European Union for the potential spread of this disease into continental Europe. And it is concerning because the disease causes huge economic impacts in agriculture, and it causes huge economic losses also. So here in this picture, you can see the symptoms of the disease. You can see this brown part in the leaf, and this happens because when the bacterium is transmitted into the plant, it grows there. It invades the xylem until it obstructs it, and then it causes water stress. So in the end, the plant dies, basically. So which are the key factors driving this epidemic? One is temperature, because the growth and survival processes inside the plant for the bacteria are driven by temperature. And then the vector distribution, because the vector is the responsible of transmitting the disease from one plant that is infected to another plant. So as I have said, there is a lot of concern. So there have been several attempts to predict the spatial distribution of the disease, indeed of Xylella fastidiosa diseases, because it can cause many diseases on many important crops like almonds, olives, and other crops. And these attempts has been done using correlative models, such as species distribution models. So I will briefly explain you what these kind of models are. Basically are machine learning models in which you try to predict the probability of presence of a given species based on some climatic variables such as temperature, precipitation, or things like that. So you have some presence data on your species. You generate some pseudo absences, and using this data set, you fit your model. And once your model is fitted, you can extrapolate the results to other zones or using climatic projections to other scenarios or things like that. But what happens is that vector-borne plant diseases are indeed emergent phenomena. So they are not basically the simple sum of the constituents of the epidemics, but they are the result of the many interactions of these among these constituents, and these interactions are usually nonlinear. So what I want to say with that is that you cannot use the distribution, the predicted distribution of the vector alone or the distribution of the bacterium alone to try to predict the distribution of the disease. So apart from this fundamental limitation that this model has, there are others like these species distribution models lack crucial epidemiological information, such as disease dynamics. There's no disease dynamics there. And as correlative models, they also have limited interpretability and explainability. You can know which are the variables that are most important to predict the distribution, but you don't know why, or you don't know the interplay between these variables. So anyway, I don't want to say that these models are completely wrong or useless, anything like that. Indeed, I think that they are still useful and valuable. till now, there was the only kind of information we had about this potential spread. So it's something. And in general, I want to show this famous sentence, no, that all models are wrong, but some are useful. And this also applies to the model that I'm going to show you now. But the question that we asked ourselves was if we could do better. We thought so. So we developed this mechanistic climate-driven epidemiological model that accounts for these things here. So basically, we account for the temperature-dependent process of bacterial growth and survival inside the plants. We also take into account the climate-driven vector spatial distribution, and we merge all these, including epidemiological dynamics. Something that I really like is that our model is mechanistic and instead of purely correlative. So we can know why there is risk or not in a given zone. Well, this model was published in Communications Biology, if anyone wants to check it. And let's go to explain a little bit the model. First of all, the general idea. So we have an initial infected host population, infected plants. Then the vectors that feed on those plants are going to feed on them and are going to get infected. Once they are infected, they are going to feed on other plants and are going to transmit the disease this way. So this creates new inoculated hosts, or new exposed hosts, as we want to say. And this process, which is modulated by vector, we are going to model it through what we call the transmission layer. But then there is another process that in which these exposed hosts can develop in the end an infection and be infectious or not. This is modeled by temperature, because it's driven by this growth and survival processes that occur inside the plant. This process is going to be modeled in the model using what we call the climatic layer. So let's go into a little bit more detail into each layer. So first of all, how do we model transmission? In mathematical epidemiology, it's usually the typical form of modeling transmission is through a system of ordinary differential equations, which are called compartmental models. So we developed a general and simple compartmental model for vector-borne plant diseases in general. And so that we start with the fundamental parameters of the disease. Then we applied some approximations, some reductions, so that we could reduce our original model to a very simple model, which is the SIR model that maybe you know it. And the good thing is that the whole dynamics in this model is characterized by a single parameter, which would be the basic reproductive number, R0, that the concept probably you will know because of COVID or other things. If this number is below 1, there's no epidemic, and if it is above 1, there is an epidemic. So the good thing is that because we started with this vector plant disease model, we know how the basic reproductive number, our single parameter of the simple model, relates to the fundamental parameters of the disease. In particular, something that we are going to use is that the basic reproductive number is proportional to the vector abundance in the system, no? So the number of vectors. So if you know the vector spatial distribution in a given zone in which you want to model the disease, you can use a given value of the basic reproduction number that is different in each cell, depending on this spatial distribution of the vector. Okay, so this is how we model transmission. And now let's see how do we model the infection of the plant. So I'm going to split this into two subparts. One is the growth process and then the survival process. So the growth process occurs surprise in the growing season. So this comprises from April to October, more or less. And during this season, the bacteria can grow inside the plant. So to model this process, we're going to use the bacterium growth rate that depends on temperature. You have here the data. We can feed this data, and we can use this fitted function to develop a metric that is going to model bacterial growth inside the plant. So basically, what you do is very simple. During this period, you know the temperature at each hour. Then you add up the contribution to the growth. No? You add the growth. And you can show that this metric in the end of the period is related to the total growth of the bacteria in the same period. So in this way, you can model the bacterial load of the plant. And in principle, the more bacteria there is inside a plant, the more probable should be that the plant develops an infection or it shows symptoms. You can say it in both ways. So what we did later was to perform an experiment in which we inoculated the plants with the bacterium. We tracked the symptoms over time and we recorded the temperature. In this way, you can basically relate this metric that we built with the probability of developing symptoms, which is basically the probability of becoming infected during the growing season. Okay, but then there is the winter season that is also called curing season. And what happens in this disease, for instance, it can be observed very well in the United States, is that during the growing season, some infections appear in northern USA, but after the winter season, these infections disappear. Basically, the plants are cured, and this is because of cold temperature. So what it has been observed is that Priest disease incidence depends on the average minimum temperature of the coldest month. But in principle, what we think is that there is a killing rate or a death rate for the bacterium that depends on let's say, cold temperatures, but this data is not available. However, there is a previous work that tries to use, with a given definition of cold degree days, that would be what you would obtain if you fitted this data and so on, to infer this kind of thing. So we use this definition of cold degree days, but then you still have to relate cold accumulation to probability of recovery. So again, there is no any experiments or any data to do this properly, but still what we did, the only thing we could do was to relate this cold accumulation, this cold degree days, with this metric, the average minimum temperature of the coldest month, because we know how it's related to pre-disease incidence. So in this way, we could build this blue curve that relates cold accumulation to probability of recovery, which is the inverse of probability of chronic infection in our model. So in the end, we can merge all these layers and build the final model, which is very easy. So the infected population at time t is given by the infected population at the previous time step multiplied by this exponential, which this would be the transmission layer. And this gives you the new number of inoculated hosts. Then you have to know if these hosts are going to develop an infection or not based on temperature. Here you multiply by the climatic layer. So basically the first factor would be the growth process and the proportion. Then you know the proportion of those inoculations that have developed an infection during the growing season. And then multiplying by this second factor would be the winter season. So you see if these infections remain or they get cured. However, we don't want to model incidence. What we want to model is the risk of establishment of the disease. So we want to know if in a given place the disease, so the infected population would grow or would decrease. So what we define as risk index is the effective growth rate of this first equation. I didn't say it, but we are using real data all the time. So temperatures are the ones that they are. So this growth is not a perfect exponential, it's noisy. So we are interested on this effective growth rate. So if this growth rate is negative, it means that there is a general exponential decrease of the infected population, and then there is no risk there. And if this risk index is positive, it means that you have an exponential increase of the infected population, and then there is risk. Furthermore, we bound this growth until the maximum growth, theoretical growth, that would happen under optimal climatic conditions when the climatic layer is basically 1. Then you can differentiate different impact zones that, for instance, if you grow at the maximum growth rate, the spread is going to be very fast, but if you grow at half of this growth rate, it will be, the impact would be lower. So we can differentiate different impact zones. Okay, so up to here we have built a model, but now we have to calibrate the model. So the only free parameter in our model is the basic reproductive number. We had a lot of data in the US of the presence and absence of this disease for different years and locations. So we could run our model for those years in those locations using real temperature data. And in this way, by just testing different values for the basic reproduction number, you can obtain the best choice. No? That would be R0 equal 8. Then for Europe, the situation is a little bit different because in the USA, there are many vectors that can transmit the disease, like 8 or so, but in Europe, only 1. So the basic reproduction number should be different, indeed should be lower, but we do not have this kind of nice data to do this kind of analysis. So we had to do another thing. What we have in Mallorca, the only data that we have in Europe is this data from the almond leaf spore disease in Mallorca, which is caused by the same bacteria and transmitted by the same vector, but affects another host, that is almonds in this case. So by fitting this curve, you obtain the basic reproductive number of this almond leaf spore disease. And then you make an assumption. You assume that the infectivity rate is going to be similar. It's not going to change. And you account for the change on the mortality rate that is known. Just by changing this mortality rate, you obtain R0 equals 5, which is lower than the one in USA, so it makes sense. And the proportion to which is lower also makes sense biologically. So we think it's a good guess. And we proceed to apply our model to different zones in the world. What we observe here is that, well, we use what we call the homogeneous vector distribution. Basically, we use the same value of R0 in each of the cells for each figure. For US, you use the R0 equals 8, and for the rest of the world, R0 equals 5 as in Europe. So we are assuming that there is vector everywhere and the same quantity. So what we observe is that all places currently affected by pest disease are classified as rich zones, but we also observe that there are other places in which the disease is not present and is also classified as a risk zone. This is not bad because it can happen that the conditions are good for the disease to spread, but if you don't have an introduction, the disease is not there. However, we also know that there are places here in which there is no vector there, so indeed there is no risk. So in Europe, we have this spatial distribution of the vector, so we can apply it in our model and then change this basic reproduction number for each cell. It will be different. What we see is obviously that the risk is downscaled. So in the zones in which you don't have the vector, then the risk is 0. And still, the remarkable thing is that all current affected zones are still identified as risk zones. So we can say that the model is very precise. And well, we have all these results and more. Here you can see the risk only in vineyards in Europe. And all this can be checked in a webpage that we developed in an interactive mode, which is, yeah, cooler. So just to conclude with this part, a couple of things. First of all, that the approach is quite general, so it could be applied in principle to other plant diseases. And something that is interesting also is that we observe that the risk in southwestern France is increasing. You can see it in the video here. And this region is an important wine growing region. So this is an important result. And as further work, we thought about combining our model with climate change scenarios to try to predict the risk in the future. And this is what we are doing with Jose and Mayale, precisely. And I'm going to show you just some results, and that's it. So what's new? What I said, that we integrate this model with climatic change scenarios, but the model is the same. And we include an analysis of impact or potential impact on wine protected designations of origin that I think it's also interesting to check. So some results here. Here you can see the suitability, the current suitability levels for the bacteria and for the vector in Europe. And then you can see the change for different warming levels. What we observed in general is that there is an increase of the bacterium suitability, but there is a decrease of the vector suitability. So then the question here is, what is going to happen with risk? Is going to be an increase of risk or a decrease? And this is what I was saying at the beginning, that you cannot do a simple overlap of these two, or how do you do it? No, but our model is what our model does indeed in a way that we think it's appropriate, including disease dynamics and so on. And then we just project this model with these different scenarios. And what we observe is that the risk increases in general in Mediterranean regions. And something I think is quite important is that we identify this +3 scenario as a tipping point for potential propagation of the disease into continental Europe, so beyond the Mediterranean region. In a +2 scenario, it's still contained in the Mediterranean. However, well, we also computed the risk velocity of the disease. So it would be how fast a given risk level, let's say, expands in space. And well, we also observed that overall Northern Europe keeps safe until a +4 scenario also. So the last results slides. Here I want to show you how the things changes when you compute the risk at a country level or at a PDO level or even at a vineyard level. So for instance, just an example, focus on France. France has, it starts with low risk and it increases. The increase in percentage is quite high, but even at a +4 scenario, only 7% of the surface is at risk. But when you check the PDO regions, you observe that half of the PDO regions are at risk. So the story changes completely, basically. And if you go to the vineyard level, you observe that 80% of the vineyard surface would be at risk. So it is really important to assess the risk at the scale that you have to do it. In this case, PDO regions or vineyards or something like that, not at the country level. Well, yeah, just to conclude, what I think is most important is this identification of the +3 scenario as a tipping point for propagation beyond Mediterranean zones, and that the overall risk is downscaled compared to correlative methods. These species distribution methods have been used also with climate change scenarios, and they project higher risks, for instance, in Northern Europe. And we say that there is no risk there, and we can answer why, if our model is correct, obviously. And this is because bacterial survival is, in essence, very difficult in Northern Europe because there are high CDD values, which means that there is a strong component of the winter curing. So it could happen that you introduce an infected plant, the vector is there, so it could be propagated. It could be that new plants are inoculated. Even in the growing season, the bacteria could grow inside the plants and generate symptoms or whatever. But in principle, if our model is correct, after the winter season, there would be this winter curing effect, and all these plants would be cured. Then the transmission change, in principle, is stopped, and there shouldn't be risk there. Again, the results are, if anyone is curious, you can check some of these results in the web page that are interactive and so on. It's quite nice. And yeah, I want to thank you all for your attention. And if you have any questions, just feel free to ask me. Yeah. This one. Yeah, this in particular is because of the, you can see it here, because of the change in the vector. The vector, because the suitability of the bacterium is going to increase in general, but there is going to be a decrease of the vector. And in these zones, the vector was not very present already. So in these zones, you don't have, the vector cannot live very well. And we think that in the future, it's going to be worse for the vector because it's going to get drier. Yeah. Because this part, Mayalen did it, the species distribution model for the vector, so she knows more, but so exactly. 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 Yeah. So indeed, for the vector, we use not only temperature, but precipitation or humidity, yeah, other factors. So, and this, the quantity of the vector, if the quantity of the vector changes, your R0 for this cell changes. So if R0 is below 1, for instance, for sure, there is no risk. As if R0, imagine it's 2. Ah, vale, vale. Vale. Bueno, the next one. And yeah, if R0 is 2, for instance, then you have to take into account the temperature of the this growing side. So there are a lot of things, but yeah, the vector affects the exponential increase if it's faster or yeah, exactly, exactly. Exactly. The question if, is if we compute if each grid cell is independent of each other, and the answer is yes. So we are using fixed cells that are like 81 square kilometers. So the assumption would be that there is no transmission from that one infected vector is not going to travel to the next cell and produce an outbreak there. But anyway, for this particular model, it's not important because what we'll do is to introduce an initial infection in all the cells. And after 7 years, we see that is the typical time scale of the disease. We see if it has increased or not. Then we move 1 year, introduce infections again, and we do so. So it's not really, really important because we are forcing the introduction. But yeah, in a model with more resolution, it could be interesting to see how the spread could also. Mhm. 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 We don't know. I mean, so the question, bueno, finish the question now. Hmm, exactly. Mhm. Sure, sure, yeah. So the question is this can be used for as a prevention tool in case you want to invest in building vineyards. So yeah, yeah. I mean, and for instance, so I also like to use this homogeneous vector distribution because here you're assuming the vector is everywhere. So you can see in which places, for instance, southern Spain, there is a lot of risk if the vector is there. But then you see that because the vector is not there, there is no risk. But it could happen also that another type of vector came, or another insect that can also transmit the disease, and it is okay with drier uh, climate. Then the risk would explode there because you are saying there is no risk because there is not the vector only. So it's interesting that our model can, because we know how it works, we can split it into different, yeah. And but also, yeah, and also the just as a level of, the bacteria can live very well there, and we also have vectors. So, yeah. But in principle, in the future, it should decrease because it's getting drier. Exactly. Easy. Yeah. Yeah. Hmm. Yeah. Or the, yeah. Easy. Yeah. Indeed, for instance, now in Mallorca, if you go there, in the airport, you can see warnings of you cannot trade plants because of Xylella. In Europe, there is this huge concern, and in those places, there is a strict quarantine measures and this kind of stuff. Yeah, but the good thing is, for instance, I don't know. Here we are assuming that the vector is everywhere in Europe with this R0 equals 5. And I don't know, in Germany, you shouldn't worry that much. I mean, if our model is right and so on, that it's very precise, blah, blah, but I don't know. It could happen that something is not well done. I don't know. But in principle, it is a cold climate, northern Europe, that yeah, the bacteria would be killed in the winter. Yeah. Thank you. Bye bye.